Alright, so the past few weeks I've been researching gene editing in human embryos. So genome editing is a method where scientists change the DNA of an organism. They take the DNA strand and cut it at a particular area where the DNA can be removed, added, or replaced. Editing DNA can drastically affect physical traits like hair color or disease risks, generally used to investigate different diseases that can affect humans and potentially treat them. But now technology is changing and advancing. Scientists have found a way to gene edit human embryos. So the first gene editing in human embryos took place in April 2015 by a group of Chinese biologists who reported the first documented experiment where they edited out genetic diseases in a human embryo. They attempted to remove an inherited, potential fatal blood disorder. They injected 86 non-viable human embryos with the gene editing system CRISPR/Cas9. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which we learned that in chapter 19, I believe. And so this just kind of shows the steps in this genome editor repair. Or in simpler terms, the sequence of DNA bases is targeted. Guide RNA locates and binds to the targeted DNA sequence. The Cas9 enzyme binds to gRNA and cuts both strands of DNA. And if it doesn't bind and cut, then the gene is left as it is, silencing it. So the experiment that was done is they used the tool to delete a faulty gene and replace it with one that produces normal blood cells. The editing worked for only 4 of the 86 embryos and created numerous unintentional mutations. Because of the mutations, scientists are wary of using this editor in humans. So it took researchers and governments by surprise and set off a global discussion about the legal and ethical implications of the research. However, this didn't really stop biologists from experimenting with the CRISPR/Cas9 editor tool. And up at the top, this just kind of shows normal gene sequence and mutated gene sequence in humans. And another kind of study that happened was a license to edit. In Britain, a team of researchers at the Francis Crick Institute in London were granted a license to edit the genes of human IVF embryos. The researchers said that they could potentially provide vital details of how genes work in embryos less than 6 days old, which could ultimately help to improve the chances of a successful pregnancy on women undergoing IVF treatment. Professor Lavelle Batch from the Francis Crick Institute said that a doctor studying with CRISPR-Cas9 will address the role of the specific genes in determining the development of early human embryo in the first days after fertilization. This work could potentially inform the selection of healthier IVF embryos for implantation, and it could also help identify mutations that could lead to miscarriages. There's another study going on as well, a 3-parent embryo study, and just a little quote: "The idea of giving the best to our children and helping them be their best has long been a quest for parents. Today, the quest for the perfect child has turned to genetic manipulation. The ability to manufacture a designer baby has recently taken a significant step closer to reality." So 3-parent babies was originally a way to treat mitochondrial genetic defects. These defects can be severe and potentially life-threatening. Mutations in the mitochondrial DNA can lead to severe diseases that don't have a cure. And this just kind of shows the mitochondrial DNA and some of the inheritable traits and diseases. So a mitochondrial genetic disease has never been treated. The embryos are now being treated with triparental contributions in hopes that the humans will not possess or pass on the genetic mutations. The 3-parent embryos are seen as clones since the nucleus is what is being transferred. So all of these experiments could lead to a major impact on society, for the good or the bad. And this just kind of shows the three-parent babies and how there's the mother egg and the donor's egg with the sperm. And technology is constantly changing and advancing the way we live and learn. Experiments today have led to new technologies and achievements to benefit the human population. Recent studies of gene editing in human embryos have a crucial impact on society. Although some negative effects may occur from gene editing, there are potential pros to this major advancement in technology. And here are my references.